Ladies and gentlemen, welcome. I'm Anne Greenoff and I'm Head of School of Medicine here at King's College London, and it gives me great pleasure to welcome you to the second inaugural lecture in our current series, and this lecture will be given by Professor Chris Hammond. Professor Hammond was appointed to the Frost Chair of Ophthalmology in 2011. The Frost Charitable Trust was created by Sir Stephen Miller and Thomas Frost in 1966 to provide grants and scholarships to universities and hospitals for research into eye disease. The Trust established the Frost Chair in Ophthalmology at St Thomas Hospital in 1990, and we are extremely grateful to them for their support of such an excellent clinical scientist as Chris Hammond. I'm delighted that one of the trustees, Liz Graham, and the ex-chair of the trustees, Michael Sanders, is with us tonight for this inaugural lecture. Professor Hammond undertook his undergraduate education at the Universities of Cambridge and Oxford and was awarded an MD by the University of London. He undertook his clinical training in Addenbrooke's, St Thomas and Moorfield Eye Hospital. Before being awarded the chair, he was a consultant ophthalmologist at the Princess Royal University Hospital, Orpington, and an NIHR Senior Research Fellow. His research has focused on the genetics of eye disorders using twin studies. Indeed, he's measured quantitative healthy variation traits in up to 5,000 twins. I am delighted to ask Professor Hammond to deliver his inaugural lecture entitled A Twin Tale, What Makes You Myopic? Chris.
Thank you, Anne. Well, thank you, Anne, very much for those kind words. And I too would like to pay my tribute to the Frost Charitable Trust who support the chair and for their generous support of King's College London and St Thomas's Hospital Eye Department. I'm going to start by paying a short tribute to the many clinicians who trained me in my clinical training. By necessity, this sort of lecture is mainly a research lecture, but I'd like to name a few very special people who were involved in my training. I started my ophthalmology career in Brighton, and Alec Harden's undimmed enthusiasm throughout his career, and Sarah Vickers' inspiration to do paediatric ophthalmology and strabismus, my subspecialty, were instrumental in moving my career forward. I went to Cambridge and worked with Professor Tony Moore, who again is one of the great names in ophthalmology today, and was very privileged to work with him. I went as a registrar to the Western Eye Hospital, where I worked with James Atchison, a neuro-ophthalmologist who inspired me again to continue in this subspecialty and taught me that a sense of humour on all occasions is a very valuable thing to have. Then I went as a senior registrar to St Thomas's and have trained with really some of the great names. Michael Sanders, who I'm privileged to be here tonight, one of the eminent neuro-ophthalmologists, not in the UK but in the world. Professor David Spalton, who's recently retired from St Thomas's, John Schilling, Liz Graham and Miles Stanford, all of whom taught me much of what I know. And finally, a tribute to my colleagues at my previous hospital, the Princess Royal University Hospital in Bromley. David True and the gang there were the best of colleagues to work with, and I've learned so much from all of those and more. But I'm going to start my lecture, A Twin Tale, What Makes You Myopic? And I'm going to start with three short twin tales. The first twin tale, here we have a picture of young blonde twins, and some of you may realize that actually the pair of twins is me and my twin brother Nick. And guess which one I am? How many of you can guess, I wonder? I'm on the left-hand side. And those twins were brought up in darkest Africa in Zimbabwe, and there they are at school in the library in the obligatory school photographs. I'm still on the left, just in case you don't recognize me. And finally, they reached the end of their school career and proudly sitting with their school prizes. Needless to say, both have now become myopic, short-sighted. Obviously, we were too vain to be wearing our glasses at the time, but we're both short-sighted there. And we cut forward to today, and I'm very proud that Nick is here in the audience, and for all his love and support over the years. And so here we have one twin tail. The second twin tail I'm going to mention is the Twins UK cohort, and in particular Tim Spector. Tim is the director of the Department of Twin Research at King's College London, and has been my mentor, and I really wouldn't be here without all his support and encouragement. Tim has been instrumental in my career, and I wish to pay him the fullest tribute. He founded the Twins UK cohort 20 years ago now, and it now has over 13,500 twin volunteers on its adult twin registry, of whom almost 9,000 have visited St Thomas's for various measurements, and we now have over 6,000 twins with measurements of their refractive error, that is the degree of short sight or long sight that they have. It's one of the jewels in the crown of King's College London, the Twins UK resource, one of the most phenotyped and genotyped cohorts in the world, and as many of you are aware, it's now a part of the bioresource of the Biomedical Research Centre, which GSDT and King's Health Partners run. And my career has been working on this twin cohort throughout. The third twin tail is, I'd like to pay tribute to, is Claire Gilbert, who unfortunately can't be here this evening. Claire proposed a twin eye study back in 1996-97 with Tim, and they were successful in obtaining Wellcome Trust funding, and to my great fortune they employed me to run the twin eye study, and there I have remained ever since. Claire herself is also a twin. She has a twin brother, and she's a professor at the London School of Hygiene and Tropical Medicine, where she's one of the most eminent epidemiologists in international children's eye health, and she is being eminent abroad as we speak. And I'd like to pay my respect to her for her role in mentoring my career and her support. Well, I think we should probably add something to death and taxes, and that's wearing spectacles. They're all unavoidable. If you look, this is an old slide from 1974, but it makes the point that during childhood and adolescence and up until the early 20s, increasing number of people wear glasses, and then there's a bit of a lull, and then of course middle age kicks in, and people who didn't have to wear glasses before then have to wear their reading glasses, and so almost all of us end up wearing glasses at some stage. The presbyopia we're not going to be talking about today. We're going to be talking about myopia, which is the commonest refractive error and the commonest reason why people are wearing glasses. How many in this room are myopic, I wonder? Oh my goodness. I'm straw poll, probably 60% of the audience, I'd suggest. And we'll come to discuss some of the reasons why we might all be so myopic, me included, during the course of the lecture. So I'm going to start by talking about the growth of the eye. The eye, when we're born, is about 17 millimeters long, the axial length from front to back. And most of the focusing of the eye is through the cornea and the lens of the eye. And when we're born, almost all of us are significantly long-sighted or hypermetropic. That is, rays of light from the distance are focused behind the retinal plane. Now, that's not a problem because children have an amazing ability to do what's called accommodate. They can change the shape of their lens in order to bring the focus back onto the retina so that they can see clearly. And from 1 week of age, our eye starts to grow really fairly rapidly. By 3 months of age, it's grown another millimeter on average, and by 4 months, up to about 19.5 millimeters, and by 12 months, 20 millimeters, and by 2 years, it's about 21 millimeters in size, and the cornea, the window of the eye, is now fully grown and contributes about 3/4 of the focusing power of the eye. Our eyes continue to grow, 22 by 3 years old, and the eye is now something like, at the age of 4, 95% of the adult size, 23 millimeters by about 5. And then when the eye reaches maturity, which is usually post-puberty or around about puberty, we have reached an axial length of 22 to 26, or in fact 22 to 24 normally. And we've reached a stage called emmetropia, and that is parallel rays of light are focused directly onto the retina in the resting state of the eye. And emmetropia is an amazingly powerful mechanism. Every animal species emmetropizes, and And if you put a lens in front of the eye of almost any young mammal, or in fact even birds, their eye will grow to accommodate that lens so that they reach, again, normal focus. Very powerful mechanism. But what's different between humans and the rest of the species is many of us in this room, for example, become myopic. And that doesn't really happen in the animal kingdom at all. People have really struggled to find natural myopia in animals. We can induce it by playing with their focus, but we can't actually see it naturally. But what happens in those of us who are myopic, the majority of us have what's called axial myopia, and that is that our eyeballs have grown posteriorly too far, and in fact they're now starting to resemble an egg rather than a round ball, and we have an axial length of more than 24 millimeters or longer. And at this point, the rays of the light from the distance are focused in front of the retina, and this is what we call myopia, nearsightedness or short-sightedness. And the problem is we can't change the shape of our lens to move the focus back. We have to have some help with concave lenses, be they contact lenses, spectacles, to help focus the eyes. It's not quite as simple as short-sighted or long-sighted or normal-sighted. As everything in life, there's a spectrum, and we can see, we measure the strength of your prescription or your refractive error by something called the spherical equivalent, which is measured in diopters. And you can see here, this is the population distribution of our twin cohort. We can see an awful lot of people concentrating around 0. And when we see that deviation from this normal distribution, it suggests that there's an active process going on controlling growth of the eye to try and stop it growing and keep it at that length. But sadly, many of us have slipped into this left-hand skew deviation and are myopic, and increasing numbers means more and more short-sighted, thicker and thicker spectacles. And of course, a few people remain long-sighted in this arm of the distribution. So why should we study myopia? Well, firstly, it's common, as witnessed here. At least 30% of the UK adult population has myopia, and indeed, Jugnu Rahi's work suggests as many as 40+% of 50-year-olds in the UK are myopic, so really very common. It obviously has a huge cost in terms of the economics of all that visual correction, and in the United States that's estimated at something like $5 billion a year for the correction of myopia. So it's pretty expensive. But from our point of view as a medical school, what's important is that there are complications of myopia that are sight-threatening. And in particular, retinal detachment, where the retina peels off the retina, a bit like wallpaper peeling off, retina peels off the back of the eye like wallpaper peeling off a wall, is sight-threatening and an emergency that requires surgery to fix it. And myopic maculopathy is a degeneration of the central macula of the retina, the central focusing part of the retina, that leads to loss of vision in late middle age and older people. It's also associated with glaucoma and cataract and several other eye conditions. And when we look at a forest plot of the effects, we can see that even people who are what we would classify as low myopes, they're between 1 and 3 diopters short-sighted, they have a double the risk of getting this macular degeneration associated with myopia. But if you're more than 9 diopters short-sighted, you're pretty short-sighted, you've got something like a 300-fold risk of getting this degeneration. This is on a log scale, so it's escalating fast, the risks up here. Similarly, retinal detachment. Those people who are low myopes have something like a 3 to 5-fold risk of getting a retinal detachment, but the more myopic you are, the higher the risk goes, until again, if you're more than 9 diopters, you've got a 100-fold increased risk of a retinal detachment. And so if more and more of our population are becoming myopic, we're going to be getting more and more of these sight-threatening complications in the future. As I said, it's a common disease, and in different populations, the prevalence ranges between 25 and 61%. But it varies between populations. It's increasing rapidly in some. It's associated with urbanization, education, and IQ, and we'll discuss some of these data in a moment. And most recently, we found that outdoor activity in children is protective. So if we look at the variability between populations, you can see in rural East Asian populations, this is children aged 11 to 13, there's really very little myopia. But if you go to the cities of East Asia, Hong Kong, urban Japan, Guangzhou in China, Taiwan, there's a very, very high level of myopia. We're talking 60–70% in 11 to 13-year-olds. So clearly there's something about urbanization that's causing myopia. It's also becoming more common. We have very poor epidemiological data in this country, unfortunately, but the Taiwanese have been collecting this data for a long time. And if you look at Taiwan between 1983 and 2000, the prevalence of myopia is rising dramatically, such that nowadays, if you're in Taiwan, in 2000, 80% of children reaching the age of 18 were myopic. And in fact, the latest data from Taiwan suggests that 95% of undergraduates at university in Taiwan are myopic now. And that's a real epidemic, and one that is storing up problems for the future, not least of which is that they're not just becoming short-sighted, they're becoming more short-sighted. So if we look at the degree of short sight rising between 1983 and 2000, we said that the average for the 18-year-olds in Taiwan is between 3 and 6 diopters of myopia by 2000. That suggests that, that they're already average moderate myopes, which mean many subjects are more than 6 diopters of myopic. They're highly myopic. It's not just happening in the Far East, although it's very dramatic there. These are data from the United States, the N. Haynes study, which is one of their big population-based epidemiology studies. And they've shown that the prevalence of myopia has risen from the low teens in 1971 to about 30% in 1999 to 2004. So again, a huge increase in 30 years. And there isn't any data in the UK, but in fact Katie Williams, who's just started a PhD working with our group, has looked at our twin cohort. And what we've seen is that the prevalence of myopia in the twins that I measured in 1998 and 1999 were less likely to be short-sighted than the twins who were measured in 2008 and 2009. So we have data here suggesting that even in the UK, myopia is becoming more common. One of the reasons is that so many of us in this room are short-sighted is because we're all a bit over-educated. And if you look, you can see that myopia, this is again in East Asian data, but myopia rises in proportion to the number of years that you spend at school, college, university, postgraduate degrees. So clearly, there's something about that educational close work environment that is also myopogenic, makes us short-sighted. Outdoor activity, as I said, is most recently been found to be protective of myopia. And the Sydney myopia study showed, interestingly, that even if you did a lot of near work as a child, these are 12-year-olds they studied here, if you spent a lot of time outdoors, you were much less likely to be short-sighted than if you spent less time outdoors. So again, there's something about whether it's the light, and some of the animal data suggests the much higher light levels outdoors may be protecting the eye from this growing too long and making us myopic. So on present day, send your children out to play outside if you're concerned about them becoming myopic. And some of the most convincing data is from Chinese children, comparing them in Sydney and Singapore. So same genetic background, as it were. In Sydney, 3% of 6-year-olds are myopic. In Singapore, 30% of 6-year-olds are short-sighted already. And that's at age 6. That's pretty staggering data, and one that may in part be attributable to their amazingly concentrated educational system, where they're all having extra lessons age 3 for competitive entry to school age 4. And it really is quite a hothouse there in terms of the amount of close work that they have to do. They had very similar levels of parental myopia, which is a major risk factor, similar amounts of reading between these Chinese children in Sydney and Singapore. But the big difference was that Sydney 6-year-olds spent 13 hours a week outdoors. Singapore 6-year-olds spent 3 hours a week outdoors. And so, again, data suggesting that this may be quite an important influence in myopia. So why am I, and why is my twin myopic, or was my twin myopic? He's had his laser surgery so that he isn't myopic anymore. But the most important risk factor in every study is parental myopia. If your parents are short-sighted, you're much more likely to be short-sighted. Well, neither of my parents were myopic, What about lack of outdoor activity? Well, I was brought up in really the outdoors of Zimbabwe, wide open spaces, outdoors all day. I failed that one. I had a very rural upbringing, but clearly the fact that I'm wearing this rather luminescent gown suggests I'm a bit overeducated, and maybe that's got something to do with why I'm myopic. But what about the genetics, if I've told you there's a lot of importance about the environment? Well, when we're looking at the genetics of complex traits like refractive error, we first want to know, is there familial aggregation? And the problem with family studies is almost every disease runs in the family. Let's take obesity as an example. Is it because they all eat the same food, or is it because they share the same genes? And this is where twin studies come into their own, because they've been described as the perfect natural experiment to separate nature from nurture. And that is because the identical twins share exactly the same genome, 100% of their shared genes, but they also share the same family environment. Whereas the non-identical twins share on average only 50% of their genes, and they are also, however, brought up in the same family environment as each other. So if these two are more similar to each other than these two, that suggests that genetic factors are important. So we did a heritability study of refractive error, and I published that in 2001, showing that genes are really very important in determining where you are in the spectrum of refractive error in the population. The original study of 500 pairs of twins was probably not powerful enough to look for shared environmental factors, with common environmental factors within a family. And so I'm going to actually show you the data from our study that we published in 2009. And Margarita Lopsch, who was a PhD student with us, who completed her PhD, worked on our myopia data set and published this paper with us. So she looked at over 2,300 pairs of twins, very similar numbers of monozygotic, identical, and dizygotic non-identical pairs, mean age of about 53. Majority of our cohort, the Twins UK cohort, is female. Sadly, women are much better at volunteering. Men are absolutely useless. And we performed what's called autorefraction, which means we got a machine to measure their degree of myopia or hyperopia, because clearly, if I was testing them and I see a pair of identical twins before me, I'm much more likely to make them a bit identical, their refraction. So the autorefractor doesn't know or care. And so these are the data. This is twin 1's refractive error against twin 2's refractive error. And you can see here for the identical twins, they're really very highly correlated. Correlation coefficient of 0.8 is extremely high for any biological variable between siblings. And if we look at the dizygotic twins, they're still correlated. The correlation coefficient is about 0.4 to 0.5. But as you can see, there's much more scatter around that sort of 45-degree perfect agreement line. And when we do the modeling, it suggests that the heritability, that's the proportion of variation explained by genetic factors, is very high at 77%. We did manage to find some common shared family environment of around 7%. That suggests the family who reads maybe is more likely to make their children read, and therefore they're all going to be myopic together. But individual environment only accounted for 16% of the variation. So really a very strong heritability. And this has been replicated in studies all over the world with very similar results. We've also done studies not just on myopia, of some other traits, and I'm not going to dwell on those because the story tonight is about myopia, but we published a heritability of cataract in the New England Journal of Medicine, and I'd like to thank John Sparrow, who's a consultant ophthalmologist in Bristol, who again helped us on this project and taught me how to grade cataracts. We did studies on pupil size, on cortical cataract, in association with Sheila West, who's a professor at the Johns Hopkins Medical School in Baltimore. And she has become really a great friend, collaborator, and supporter of my career. And I owe her many thanks over the many years I've been working with her now. We've also done heritability studies of macular degeneration, corneal thickness, macular pigment, and Melissa Liu was an MD student who worked in our lab and did our macular pigment study. And we finally moved to some heritability studies of glaucoma, and in particular Francis Carbonaro did his PhD with me looking at a twin study of glaucoma. And again, we've shown that genetic factors are important, maybe not as highly heritable as myopia, but still important. So on. And Sam Fahy, who's now a postdoc in Nottingham, who can't be here tonight, did a study on retinal vessel caliber, retinal artery and vein widths in the retina. And finally, Jelle Verhoef is an ophthalmologist from Groningen in the Netherlands, and he's working with us and has done some dry eye studies on our twins. So we've sort of run a fairly full range of diseases, but we're going to come back now to the myopia story. If I tell you that genes are important in myopia, then clearly you're going to say to me, well, what genes are involved in myopia? And up until a couple of years ago, we didn't know any genes that were associated with simple or common myopia. And part of the reason for that is it's been very difficult to find them. We've all got 20,000 genes in our genome. We've got 20 billion markers along, or alleles along our genome. And it's been very difficult to find which genes were associated. And up until about 7-8 years ago, we could only find rather rare alleles that conferred a very high risk of a disease or a trait that generally caused Mendelian disease, that is, diseases that were directly inherited from parent to child in a known heritable fashion. But that's when we started looking for genes for myopia. And so we started with a family-based method called whole genome linkage scanning. And what you do is place microsatellite markers, about 400 of them along the genome, or nowadays we use single nucleotide polymorphisms, or SNPs, which we'll hear a bit more about later, and generated quantitative trait loci and looked at LOD scores. And then and it's a bit like finding a needle in a haystack. Your genome, if you imagine, is a whole field full of haystacks. When we do linkage studies, we could narrow it down to a haystack, but there were still quite a few needles hidden within that haystack, and we had to try and find them out. And what we did was candidate gene studies. We'd look at the possible genes in that haystack and decide which genes might be suitable, and then try and genotype them, fine map them, or do association studies to try and identify the genes. So we published the first genome-wide linkage scan for myopia back in 2004. And as a researcher, when you get this LOD score of more than 3, you're terribly happy because this is highly significant. And we got reported 4 loci with LOD scores of more than 4. And one of the problems of why linkage studies haven't found many genes for common complex diseases is that they've been very difficult to replicate. And we were fortunate enough to be able to replicate one of our loci on chromosome 3q with another subset of our twins' data. And Toby Andrew, who previously worked with us at the Department of Twin Research, worked with us to report a fine mapping study of our chromosome 3q locus. I'm not going to go into too much detail, but what we did is put a whole bunch of markers along the genome in the area that we felt was harboring a myopia gene or more than one gene. And Toby did a fairly fancy multi-level, multi-locus modeling method, and we found some evidence of association in a gene region called the MFN1 gene. And this is very interesting because it raised the possibility of mitochondria, which are the little packets of energy in our cells, as being maybe involved in the etiology of myopia. And what was very interesting is this MFN1 gene interacts with the OPA1 gene, which is one we know a lot about because it causes a familial form of sight loss called dominant optic atrophy, where the optic nerve degenerates. And another one of the genes we found in our area called PARL also interacts with the OPA1 gene. And Toby has taken on this research, and he's looking at both for myopia and also in metabolic phenotypes as well. As part of the genetics work on myopia that I've been involved with, I've been privileged to work with the 1958 British birth cohort. This is a cohort that everyone born in the UK in a single week in March 1958 has been followed up. And we've published 2 papers with data just from the 1958 cohort. And it's a great pleasure to see Jugnu Rahi in the audience. Jugnu is Professor of Epidemiology at the Institute of Child Health at University College London, and not only is the most tip-top, fantastic epidemiologist you'll ever meet, methodologically absolutely precise and clear, but she's a great collaborator and a great friend, and I'm privileged to work with her. But so we've started tiptoeing along the route to finding genes for myopia. We also, in fact, found a gene, the first gene for age-related cortical cataract, the EFA2 gene, with some of our American collaborators along the way. But we then hit the genome-wide association studies era, and this is from the mid-2000s. And finally, the technology had caught up, and we were able to place hundreds of thousands of markers along the genome for each individual and measure them and analyze them. And so we've been doing quantitative GWAS studies for myopia, for refractive error in our twins population. And really there's been a revolution in the genetics of many diseases over the last 7 years, ever since the first GWAS was published in mid-2000. And what genome-wide association studies can do is can look for common variants, that is, variations in the genome that affect more than 5% of the population, and see whether they're associated with common diseases. Generally, we're finding very low effect sizes of these common variants, but because they're very common, they can have a big impact on a disease. And in fact, the one major example of a high effect common variant is age-related macular degeneration, the first GWAS that was published. And 40% of us in this room carry a high-risk allele for age-related macular degeneration, which really has huge effect sizes, 3- or 7-fold if you're homozygous for the risk allele of getting AMD when you get older. So that's really, in fact, proved to be the exception to the rule. Most of the common diseases, we're finding very small effect sizes. When we're doing a genome-wide association study, as you know, your chromosome contains your DNA, and within the DNA there are areas of genes which code for proteins that are used in our body. And along the genome, we have all these alleles or markers which the nucleotides form in pairs here, and we can use these as single nucleotide polymorphisms, markers of genetic variation in our DNA. And as I said, we could put hundreds of thousands. In fact, the latest chips, you can put 2.5 million markers along each person's genome and measure them. And so they've been all the rage in genetics over the last few years. And this is one of our early attempts to find genes for myopia. And the problem is you're doing 500,000 experiments per person. You need to be pretty sure that you're not generating any false positive results. Most science gets by on saying a 1 in 20 chance that this isn't a false positive result. If we accepted a 1 in 20 chance, we would have tens of thousands of false positive results for each of our studies. So we have to set the bar very high. And in fact, a p-value of times 10 to the minus 8 to be genome-wide significance, which our early myopia study didn't show. But when we got the numbers up to 4,000 twins with genome-wide study, we finally were able to find genes that were associated with myopia. So these are the technical details of our genome-wide scan that we performed on our twins. And when you think that when we started the GWAS era, it was around about $2,000 a person to have your genome scanned. This was all pretty expensive research. When you do a genome-wide association scan, you generate this thing called a Manhattan plot. And it's called a Manhattan plot because it's said to resemble the skyline of Manhattan at night. And of course, what you pray as a scientist is that you're going to get a few Empire State Buildings because they are going to be highly significant. And on bended knee, we were grateful to see that we had one genome-wide hit on our myopia scan on chromosome 15. And when we look at it in a little bit more detail, we can see there's a whole range of SNPs, which again is reassuring that you've probably got a real result there. And when we show the association plot, we can see a whole bunch of markers here in this area between areas of what are called recombination, these blue lines. And they overlap the promoter of a gene called RAS-GRF1. And we were very excited by this. The odds ratios, as I mentioned earlier, are pretty low, so that your odds of being myopic as opposed to being normal-sighted, if you've got one of these risk alleles, is only about 1.2-fold. If we compare your odds of being myopic versus being hyperopic, being long-sighted, with a homozygote, we can get it up to about 1.8 risk. But clearly, these are fairly low risk and much similar to most genome-wide association studies that have reported. But because of all this huge statistical energy that you put into these data, you do need to make sure they're real, these results, and so it's very important to replicate them. And we were lucky to work with the Rotterdam Eye Study, who have over 9,000 people with genome-wide association study and myopia data, the ERF study, which is a study from the Netherlands as well, the 1958 British birth cohort, 1,000 subjects from a Drugnew study, and the Australian twin study, also of 1,000 people. And when we look at our top marker, our SNP marker, for each of those cohorts, we can see they're all heading towards myopia and with varying degrees. But when we combine them in a study of over 17,000 people, we see a very highly significant result. And basically, if you've got one of these risk alleles, your spectacles prescription is more myopic by about 0.15 diopters, which doesn't sound a lot, but is highly significant. The RAS-GRF1 gene is a gene whose protein is expressed in the retina, and that's very reassuring again, because we think that the signaling that tells the eye to grow and become myopic is generated by the retina. And indeed, animal studies, you can cut their optic nerve and they're not connected to their brain anymore. The eye will still grow in response to you placing a lens in front of the eye, for example. Interestingly, there is a knockout mouse for this gene, and they have normal brain structure, but they perform worse in long-term memory tests. Now, how you test a mouse's long-term memory is kind of interesting, but it's very interesting from our point of view in that we know that myopia is associated with education and, in fact, also with IQ, and so maybe this is a possible shared genetic effect. The retina looks normal in these knockout mouse, but it definitely doesn't work normally. It is impaired when you don't have any of this RAS-GRF protein in your retina. So this was very interesting, and it created quite a stir when we published this paper. And from all over the world, it's quite interesting seeing your name written in all sorts of different languages and dialects. But we got a fair amount of interest in this first gene for myopia. As ever, the Daily Mail managed to get it a bit wrong and found the gene that causes short sight. And as I've explained, this is really just one very small gene of probably many that is involved in your refractive error. They also said that I said that the condition could be halted by eye drops, which is, again, slightly worrying. But it was only me trying to tell them that we weren't going to be doing gene therapy on 30% of the UK population, I think that made them decide there was an eye drop rather than a gene therapy treatment that we were going to give based on our finding. But actually what we're hoping to do with these sort of finding these genes is actually finding pathways that we can influence to try and influence the risk of myopia either developing or, more importantly, progressing. So at the same time, so we published this in Nature Genetics, and I'm very grateful to Piro Heisi, who's the postdoctoral genetic statistician who works in our group, who sadly is not here today at the American Society of Human Genetics, where he's talking today on some of our work. But we published this paper, and in the same edition of Nature Genetics, the Rotterdam cohort published their genome-wide association scan that our twin data had replicated their findings. And Caroline Klaver runs the Rotterdam Eye Study, and we again have developed very successful and happy collaboration over now several years. They also found one hit, and that was interestingly also on chromosome 15, but wasn't the same as ours. And you can see here their Manhattan plot. And when we look at the association plot, they have several genes in the vicinity of their top hit, but probably the one that is of most interest is this one called GJD2, which is a gene that again is involved in signaling within the retina and may be involved in the etiology of signals that are telling the eye to grow or not to grow when you're becoming myopic or not. So finally, I can say we have some myopia genes or some myopia candidate genes. Now, we've published quite a few genome-wide association studies in the meantime, but the two people I want to pay tribute to here are David Mackey, who's a professor of ophthalmology at the Lions Eye Institute in Perth in Western Australia, and Terry Young, who's professor at Duke University in North Carolina. Terry, David, and I put in a grant to the NIH to fund these genome-wide association studies in our twins, and were luckily successful in gaining that funding. And again, the three of us now have 10 years of collaboration and working together. David is the most amazing man, for those of you who know him. He has collected 1,700 twins of his own by essentially measuring them all at weekends. He used to fly to Brisbane, measure 50 twins' eyes in sort of cadging private practices from other ophthalmologists who weren't using their offices at the weekend, then fly back to work on Monday morning again in Melbourne, where he was at the time. He's really, truly inspirational and one of the most collaborative people you'll ever meet. And Terry Young is very well known for myopia genetics, looking at high myopia in families where it runs in an inherited fashion. And we've done some genome-wide association studies for glaucoma as well, and similarly, I'm very privileged to be working with Ananth Viswanathan, who is in the audience here today. Viz is a consultant at Moorfields Eye Hospital and is running the glaucoma genetics of the Blue Mountains Eye Study from Australia, one of the big epidemiology cohorts, and this is part of the Wellcome Trust Case Control 2 consortium. And again, we've established a very happy working relationship. And we've even published a GWAS on eye color. So where are we going next for myopia genetics? Well, the bigger is better, and so we've formed some international collaborations, and we now have 27 groups across the world pooling their data for myopia and genetics. And indeed, we've just submitted a study of 38,000 people across the world with refractive error data and genome-wide association studies to try and find some more genes, because, as I say, we need more and more numbers to get greater power. And this is our paper, which the analysis was done here at KCL. And you can see, in fact, we've now got 20 genome-wide significant hits. And some very interesting candidates on chromosome 6 here. We can see there's a potassium channel KCNQ5 and LAMA2, a gene that may be involved in cell adhesion and migration. We've got a tox gene here on chromosome 8, which is another signaling gene. Our original genes in this international collaboration, GJD2 and RAS-GRF1, are still there, highly significant. And so we're finally starting to put a picture together of the molecular mechanisms that may involve development of myopia and hopefully, therefore, one day be able to influence it. Now, it came as a bit of a shock to us. I don't know how many of you have heard of 23andMe, but 23andMe is one of these personal genomics companies. It's actually owned and run by Mrs. Google, the wife of one of the Google founders. And you can send your saliva across to America, and they'll do a genome-wide scan for you. And you can now do it for less than $300, and you can get your own genome back. And they are now doing research on all the genomes that they have in their laboratory, and it's rather wonderful research, crowdsourcing research. You get the participants to pay for the research, and you get to publish amazing papers. Anyway, it proved a bit of a shock to us, because at the same time we were analysing our data, up popped 23andMe, and they'd submitted their paper on the GWAS of 23,000 myopes and 16,000 controls for review. Now, what's really interesting is we've very carefully, scientifically measured people's exact refractive error and eliminated this person and that person if it wasn't quite right, and we've used population-based studies only so that it's all very representative of, and so on, so on. They said, are you short-sighted, and what age did you wear your glasses, as their proxies for myopia and your degree of myopia? And lo and behold, they find 18 genome-wide hits, and if I concentrate a bit further down, there's a remarkable concordance. Here we are, KCNQ5 and LAMA2, I mentioned a moment ago, tox gene, the GJD2 gene again, RAS-GRF gene again. So really, the big studies are all becoming incredibly concordant. We're all finding the same genes, which is clearly reassuring. We're all finding the right answers, and we all seem to be attacking it from different angles, but finding the same results. But really, a real interesting way of doing research now. Who needs expensive grants when you can get people to pay for it yourself? So just as a bit of fun, I sent off my DNA to 23andMe, having my curiosity having been raised by the fact that they were studying my disease. And so we've just looked at the variants in my genome, that the significant ones that we reported from our study. And what you can see here is that I do, in fact, am on the myopia risk for the majority of the variants. A few of them I'm pretty neutral, one of them I'm protected. And so, although this isn't great science, this is just an example. So probably the reason I'm myopic is that I have a genetic susceptibility on top of all that education and reading that I've been doing. And that's obviously going to be one of our great hopes of genetic epidemiology, is once we understand a bit more about the genetics, maybe we'll be able to work out how the environment interacts with those genes for those of us who become myopic. So what have the genome-wide association studies taught us? Well, they've taught us that myopia is not age-related macular degeneration, and we have many genes of small effect. Now, this isn't a problem in the sense some people think, oh, well, then you haven't found anything very useful. But we know that very important pathways can be identified that we can very successfully interfere with, even if they've got a very small effect in a genome-wide study. And the classic example of that is the statins. Many people take statins to reduce their cholesterol, but actually the HMG co-reductase enzyme, which statins interfere with, on the genome-wide studies of cholesterol and lipids, is there, but it's a pretty small hit. And so what it suggests, but we know that it's clearly a crucial enzyme and a crucial part of the pathway that blocks cholesterol being made and therefore reduces our blood cholesterol. But it took 2 studies, each with more than 4,000 people and each with more than 8,000 subjects to replicate, to find one genome-wide hit each. So this has been pretty expensive research. And we're, again, the great joy of the Twins UK cohort and indeed the Rotterdam study and some of the other population-based cohorts is because we can study so many different diseases. I just look after the eye diseases, but we're studying many other aspects, be they blood pressure, diabetes, bone health, and so on. We get a pretty good value for money from these expensive technologies. Myopia is a bit analogous to height. Height is a very strongly genetic predetermined thing. You can predict your height based on your parental height pretty accurately. And height is, again, something like 80% heritable. But when they've tried to find genes, again, they're now having to do genome-wide studies of over 100,000 people to find significant SNPs. And 40 SNPs explain about 5% of the variation. All our SNPs that we've discovered in our meta-analysis here are explaining about 3 or 4% of the variation of refractive error. So there's a whole lot of unexplained heritability that we still need to try and find why and how the genes are involved in this trait. But it isn't actually, although I've shown you my joke example of my own genome, it isn't useful for personal genomics. This is still very non-predictive. Knowing your risk profile for these genetic factors is not going to predict how myopic you are or if you're going to be myopic. So there's no point getting your children tested now to see whether it is going to predict them. But the hope again in the future is this famous promise of personalized genomic medicine, where we will have personalized medical care based on our genome, is around the corner, but not here yet, certainly for myopia. So where do we go next in our search for myopia genes? Well, what we're currently looking at is the bit in the middle here, the low-frequency variants, which happen at less than 5% of the population, but have an intermediate effect. And part of the Twins UK cohort resource is that we are having whole genome sequencing done on 2,000 of our twins, again, very new and expensive technology, which we hope to look to see whether less common variants are associated with myopia. And this is our current research team in the Department of Ophthalmology here at KCL. And Piro Heisi, who I mentioned earlier, is looking at the rare variants in myopia. He's also looking at metabolomics in eye disease and also has an interest in structural variations in the genome, which is what he's talking about in San Francisco as we speak. Abhishek Nag is a PhD student in our lab who's looking at the rare variants in glaucoma. Omar Maru is the academic clinical lecturer at King's College London, and his research is the electroretinogram, and he's measuring ERGs in our twins and also in subjects with rare genetic dystrophies to try and work out how the normal ERG works. Katie Williams recently started with us and is doing a study on the early development study, the TED study at the Institute of Psychiatry. And this is a huge cohort of 15,000 twins who've been followed up from birth and are now 18. And they've had a huge amount of educational, psychological, behavioral, cognitive factors measured, and we are now going to get their refractive error and try and see if we can work out for the first time whether, for example, the genes that are involved in refractive error are also the same genes that are involved in IQ or educational attainment, for example, or whether they share environmental risk factors rather than genetic risk factors. And Kate Yonova Doing is an MSc student working with us from Erasmus in Rotterdam and working using our data sets to look at cataract genetics and also starting some work on diet data and cataract. Christina Venturini is working on the Blue Mountains Eye Study glaucoma cohort with Viz and Jugnu, who are her supervisors, but she's based in our lab because we're all doing similar work together. And Diana Kozareva, who's the research assistant who works with us, is collecting glaucoma and cataract cases and controls from the clinics for our genetic studies and also helping with the measurement of the twins. And finally, last but not least, is Phil Saker, who's my PA, to whom I'm very grateful for her help in putting these fancy slides that move and technology that I wasn't very good at. So where do we go next? Well, there are other directions we'd like to move. Firstly, we'd like to look at the epigenetics of refractive error. Epigenomics are the heritable changes in gene expression throughout our life in development and beyond. Genes are turned on and off. They express different amounts of protein, and changes in methylation and histone modification cause genes to be turned on and off. And we are very lucky to have the discordant twin model. If one identical twin is myopic and the other twin is not, clearly something has happened along the way to make them different because they've got the same genes. And so we can look at differential methylation in these twin pairs to see whether we can identify any genes that are differentially turned on and off in those pairs. We also hope to look at the epidemiology of myopia and vision loss, and this is work that Jugnu Rahi and Phil Cumberland are leading on at the ICH, and that's part of the UK Biobank project, this huge project where 500,000 people have their DNA stored and have various measurements done across the UK, and about 120,000 of them have had their refractive error measured. So these are vast studies that we hope to be able to dissect further out, including, we would hope, the genetics of myopia using that cohort. We'd like to do an intervention study to see if we can influence the progression of myopia. Unfortunately, children who develop myopia early on almost always end up very highly short-sighted and at risk of those complications that we heard about earlier in the lecture. And so one of the areas of interest at the moment is different wavelengths of light. It seems that that blue sky we see outdoors may be better for us than the red light reflected from these chairs and my gown. And there's some interesting work going on in light wavelengths, and whether we get children to wear a tinted lens that reduce the amount of red that reached their eye. There is some interest in setting up a clinical trial to look at that. And finally, this holy grail I mentioned, can we find gene-environment interactions that may help us in the future develop population-based interventions to try and reduce the amount of myopia that develops. Well, I'm going to finally conclude with three acknowledgement slides. The first acknowledgement is the great teams that I work with. The top picture is the Department of Ophthalmology on our summer sports day at Lambeth Palace Park opposite St Thomas's Hospital. And the bottom picture is the Department of Twin Research on our summer outing down to the south coast. And I'm really very privileged and blessed to work in two such happy and harmonious teams who really are fantastically productive and make my life so fulfilled. And for all the people in the audience who are from those departments who I can't individually mention, I thank you very much indeed. And also thank in particular the fact that I've just spotted Marlies de Lange, who's come all the way from the Netherlands to be here at the lecture this evening. Thank you, Marlies, who worked with us on our cortical cataract paper and on the twins cohort. Thank you for coming. The other acknowledgement I have is the village of Alfriston, which is in Sussex, very near the Seven Sisters on the south coast, you can see here. As some of you may know, I have an interest in a tea shop there, Badger's Tea House, purveyors of the finest cakes, and highly recommend you go along to eat cake and drink tea there. Sometimes at weekends I'm let loose and allowed to help with the baking. Well, allowed, at least most weekends I'm forced to help in the tea shop. And the tea shop is run by my partner Michael, and I'd like to thank him for all the love and support that he's given. My journey here today wouldn't be half as much fun or fulfilled or organised without him. Thank you, Michael.
I now call on Professor Emile, Head of Division, to give the vote of thanks.
Well, Chris, thank you very much for a wonderful lecture. I think it's easy for all of us who are here and privileged to hear you this evening to understand why you were appointed to the Frost Chair of Ophthalmology and how very lucky we are as a division to have you join us. By his appointment, Chris became a member of the King's College London Division of Diabetes and Nutritional Sciences, and it is clear from your final slides that you are very serious about the links between ophthalmology and nutrition. Those of you who were listening will also have noted you commented upon obesity briefly and only once during the course of the lecture, and that was a great delight to us also. More seriously, I think what that lecture has demonstrated is the excitement of research that you have so clearly experienced. You've obviously been describing something you feel very passionate about, and you've also shown us the really exciting parts of research, and particularly the collaborations where you've been very generous in acknowledging the people that you've worked with throughout the course of your talk, and I think that is one of the exciting things that being in research and academe brings to us. It's also important to remember that in addition to research and clinical, one of the roles of a professor is in teaching. Indeed, some of you will remember, those of you who are old enough will remember, that the original meaning of the inaugural lecture was for the candidate for the chair to come and give his lecture to the students to see whether or not they approved of him enough for the institution to offer him the post. I think the assembled students here fully support the decision we made preemptively for them when with the Frost Foundation we appointed you to the chair. I think it's been a very exciting presentation. You've made a complex subject comprehensible to people from different disciplines within the school. You've shown us where the basic science can take us in terms of changing things for patient benefit, and it's been an exemplar of inaugural lecture, and we are delighted to have you create an ophthalmology research strand in our division. Thank you very much indeed.
Thank you.